Good evening. In this topic today we will discuss about the one of important organ, internal organ and largest gland in our body, that is the liver. What are the features? Where that location? What are the functions? What is the anatomical structure of liver? We can see it in this session. Okay, liver. So liver is important, one of important organ which is present right side of our body below the abdominal area below the diaphragm, right upper quadrant. They secrete the bile juice. It helps in already in our previous um sessions. We'll see that we will we had seen that that is the liver secretes a bile bile juice. This bile juice helps in emulsification and helps in change the medium of our digested food. Okay, so let us see some features and functions of liver is the largest gland. First one is the largest gland of our body. It is nearly 1.5 kg, 1.5 kg, right side located, right abdominal right abdominal upper quadrant below the diaphragm below the diaphragm. uh, Next, it's covered by a capsule, that is the Glison's capsule. It's a sheath. It's covered by a sheath. First, above the Glisson sheath, there is a serosa sheath is present. Below the sheath, that is the Glison's sheath covers the liver. It is dark brown color. Dark dark brown color. Liver is a dark brown color. It has five lobes Actually we can see two lobes. Main lobes are two, but again these lobes divided into multiple, that is it has five lobes. Total five lobes are present. This is dark brown color. Shape, wedge shaped wedge shaped structure. This is the largest gland, 1.5 kg weight, right abdominal cavity, um, opposite of our um, uh, that is stomach. This is stomach. Here present the liver. Okay, it has five lobes. This is the main functions. What are the functions of this is the diaphragm. What are the functions of liver helps. It has so many main functions. It secretes the bile juice, greenish yellow color, greenish yellow colored uh, liquid, that is the greenish, that bile juice. Hepatic cells of liver secretes the bile and stored in the gallbladder. From here they reaches into duodenum. This bile juice helps in emulsification emulsification means the change that uh, uh, fats cuts into the small structures, that is the glycerol or fatty acids. Next one is the this bile juice contains sodium salts. This helps in change the medium of food into acidic to alkaline. This is the remaining main functions are glycogenesis 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 means after digestion, excess amount of sorry carbo not carbo this is a glucose. That glucose convert into glycogen. In the form of glycogen, they are stored in the liver. This process is called glycolysis glycogenesis glycogenesis. Next one is the glycolysis. Glycolysis means whenever the blood sugar levels are falls down in our blood circulation, then we need energy, no, to function of our organs. We need energy. That energy, then at the time of uh, uh, hepatic cells, what are the glycogen, stored glycogen, glucose, they converts into by the lysis means they splitting those glycogen into glucose and send into the blood circulation. That is the glycolysis. Means converts the sugars into glycogen. Glycolysis means whenever the sugar levels are low in our blood circulation, then that glycogen converts into glucose. That is the glycolysis. Next, generation heat generation means the liver cells will generate the temperature and distribute that throughout our body equally by the blood circulation. Next one is glyco next one is storage of iron. After disintegration of uh, red blood cells. After disintegration of red blood cells means after 120 days, those are the red cells are lifespan is completed. After completion of lifespan, they destroy the red blood cells in the area of liver. Then the hemoglobin contains the iron. These iron molecules are stored in the liver cells. Next one is the formation of urea. Excess amount of amino acids. After digestion, the proteins converts into simple soluble form. That is the amino acids. These amino acids are not stored in our body. These amino acids converts into urea. They secrete through the excretion. That is the urea formation. Next one is the detoxifying. Detoxifying. After uh, GIT, all the blood vessels from the GIT, they will comes from the first they will reach us to the liver. ఈ జీర్ణ వ్యవస్థలో ఉన్న బ్లడ్ వెజల్స్ అన్ని కూడా ఎక్కడికి వస్తాయి? ఫస్ట్ లివర్స్కి వస్తాయి అన్నమాట. అంటే ఆఫ్టర్ డైజెషన్ ఆల్ ద nutrients will enter into the blood circulation. Through the blood circulation, they reaches the liver. Automatically our liver, whatever uh, toxic materials are present in our food, they detoxify like alcohol. Okay, next one is synthesis of fibrinogen. This is a protein. It helps in blood clotting, coagulation. రక్తము గట్టకట్టడానికి మనకు దెబ్బలు తగిలినప్పుడు ఆఫ్టర్ వెనెవర్ wounds ఆర్ స్కేర్స్ ఆర్ ప్రెసెంట్ దట్ అట్ ద టైమ్ ఆఫ్ ఇట్ హెల్ప్స్ ఇన్ బ్లడ్ కోయాగులేషన్. రక్తం గడ్డకట్టడానికి ఉపయోగపడుతుంది. నెక్స్ట్ హెపారిన్ synthesis of formation of heparin. This heparin prevents the blood coagulation inside the blood circulation, inside the blood vessels. మన రక్తనాళాల్లో రక్తం గడ్డ కట్టకుండా ఈ హెపారిన్ అనే ప్రోటీన్ ఉపయోగపడుతుంది. ఈ ప్రోటీన్ secretes the liver. These are the some main functions of liver: glycogenesis, glycolysis, temperature, generation, storage of iron molecule, urea formation, detoxifying, synthesis of fibrinogen and heparin. Okay, it is present in the uh, abdominal cavity, right side, upper quadrant. It covers the Gleason's capsule. It is dark brown color, 5 lobe structure, wedge shaped structure. Okay, internal structure we can see the next class. Okay, thank you.